welcome to biology class. I am Shikha Sharma from Adarsh Vidya Mandir Shankar Vidyapit, Mount Abu for class 11. Continue our topic photosynthesis in higher plants. We have already discussed about the light dependent reaction. This is cyclic and non-cyclic photophosphorylation. Now we come to the dark reaction. Dark reaction is also known as the Calvin cycle or C3 cycle. It occurs in the stroma of chloroplast. What is stroma? Stroma is the liquid present inside the chloroplast. So in this stroma, the carbon dioxide comes from the atmosphere and RUBP is the ribulose 1,5-biphosphate. It is a 5-carbon compound. It is already present in the stroma. So the 6 molecules of RUBP react with the 6 molecules of carbon dioxide in the presence of Rubisco enzyme. Rubisco enzyme is the most abundant enzyme in the world. So in the presence of Rubisco enzyme, these two, carbon dioxide and RUBP, react with each other and forms PGA, phosphoglyceric acid. The first stable compound of this cycle, Calvin cycle, is PGA, and PGA is a 3-carbon compound. That's why it is known as the C3 cycle. This is the first stable compound. Now, what happens? Phosphoglyceric acid consumes 12 molecules of ATP, adenosine triphosphate. Then it will convert into the 1,3-PGA, 1,3-diphosphoglyceric acid. Then 1,3-diphosphoglyceric acid consumes 12 molecules of NADP acid and convert them into NADP and convert into the 3-PGAL, glyceraldehyde 3-phosphate. So the question arises, how the ATP and NADP molecules are come to the Calvin cycle. Actually, ATP and NADP molecules are the product of light reaction. We have already discussed. So this cycle, this Calvin cycle, is continued with the help of ATP and NADPH2, which are the product of light reaction. Now, these 3-PGL, this is the glyceraldehyde 3-phosphate. 3-phosphate means on the third position, the phosphate is present. 12 molecules of PGL are formed during this. Then, out of 12 molecules, 2 molecules are used for the production of 1 molecule of glucose. And glucose is the product of photosynthesis. Glucose is the product of dark reaction. So, 2 molecules of PGA are consumed for 1 molecule of glucose, and remaining 10 molecules of glyceraldehyde 3-phosphate again produced RUBP, ribulose 1,5-biphosphate, with the help of 6 ATP molecules. This is known as the Calvin cycle or C3 cycle. But in C4 plants, what are C4 plants? C4 plants are the xerophytic plants, desert plants. They have the Grange anatomy. They have Grange anatomy in their leaves. So, what is the Grange anatomy? Actually, the upper epidermis and lower epidermis in the leaf structure. Between the upper and lower epidermis, the mesophyll cells are present. These are the mesophyll cells, and in the center, xylem and phloem, the complex permanent tissue or conductive tissue, or we can say the vascular bundles. Vascular bundles are present, and these vascular bundles are surrounded by the bundle sheath cells. These bundle sheath cells are the special cells which are present only in the C4 plants. This is known as the Grange anatomy. So in C4 plant, Hatch-like pathway is happened at the place of C3 cycle. Now what is the Hatch-like pathway or C4 cycle? You can see this is the mesophyll cell, and mesophyll cell is attached with the bundle sheath cell. Okay. In the center, xylem and phloem are present, conductive tissue, and conductive tissue are surrounded by the bundle sheath cell, and bundle sheath cells are surrounded by the mesophyll cells. So this is the mesophyll cell. In the mesophyll cells, carbon dioxide enters from the atmosphere, and in the mesophyll cell, this carbon dioxide is converted into the bicarbonate ion, that is HCO3. Then bicarbonate ion react with phosphoenol pyruvate, which is already present in the cytoplasm of mesophyll cell. Then PEP react with bicarbonate ion and it convert into the OAA, that is oxaloacetic acid. C4 acid, the first stable compound of C4 cycle is oxaloacetic acid, which is 4-carbon acid. Now oxaloacetic acid enters into the bundle sheet cell. It transports into the bundle sheet cell. Then in bundle sheet cell, decarboxylation of oxaloacetic acid is happened. Means one molecule of carbon dioxide is removed from it and it will convert into the C3 acid. What is C3 acid? C3 acid is the PGA, phosphoglyceric acid. Then PGA is again entered into the mesophyll cell and the Calvin cycle is continued during this and glucose is produced and again PEP is formed. So this is the Hair-Chai-Slake path. Now we will come to the last point of this chapter, that is photorespiration. Photorespiration, or we can say the C2 cycle or glycolate pathway. Actually it is a wasteful process in C3 plants and it is enhanced by the bright light, high temperature, high oxygen concentration and low carbon dioxide concentration. So it happens only in C3 plants during the daytime approximate 12 or 1 o'clock during afternoon. So what happens in this wasteful process? In C3 plants, chloroplasts are present, peroxisomes are also present and mitochondria is present. So this cycle, C2 cycle is happening. What is the first step? The first step is in the chloroplast. RUBP is present in the stroma and RUBP react with carbon dioxide and convert into the PGA. It is a simple process we have studied. But what happens in the photorespiration? This RUBP react with oxygen at the place of carbon dioxide. It react with oxygen and convert into the phosphoglycolic acid. And this phosphoglycolic acid is transferred into the peroxisomes. From chloroplast is transferred into the peroxisomes and phosphoglycolic acid it convert into glycolic acid. Then glycolic acid convert into glycine. Glycine is the amino acid and this glycine is entered into the mitochondria. What is mitochondria? It is the powerhouse of the cell. So glycine amino acid is converted into serine amino acid in the mitochondria. Then from the mitochondria, this serine is goes into the cytoplasm and make PGA, phosphoglyceric acid. Then phosphoglyceric acid converted into the carbohydrate, the glucose molecule. But during this, you can see at the place of carbon dioxide, RUBP, ribulose biphosphate, is react with oxygen. That's why it is known as the photorespiration. Because during the respiration process, oxygen is consumed. So, in this process, at the high concentration of oxygen, RUBP react with oxygen because the carbon dioxide concentration is low at this time. So, directly it can't convert into the glucose and it goes through the glycine and serine amino acid. So, and in the C4 plants, this photorespiration is absent. Now it is all about the photosynthesis in higher plants. Thank you.